Hello students, welcome to academia. Today I will discuss about the museum technique in histology. What is museum? Generally, all of you know that the museum is a place to exhibit objects, an instrument of research and a platform for personal teaching. But what the museum technique means in histology. All teaching hospitals and colleges of pathology have museums which serve many functions like permanent exhibition of common specimen for students, illustrating specimens of rarity, permanent source of histological material, and for gross and microscopic photography. So these purposes are served by the museum techniques. In today's video, we will see that what are the basic techniques of the museum and how the specimens are kept in the museum. So first we see that how the specimens are displayed in the museum technique. Number one, the specimens arranged in divisions on steel racks in a systematic way. It should have a code number. Various aspects of a disease displayed at one place. Fresh specimens displayed after grossing, placed at the entrance of the museum with all available information about the patient. And also, the learner can personally handle and feel the specimen. So this is about the recent specimen, and some specimens are of fluid in nature like the urine sample, CSF, blood samples. These are also displayed in the museum. Now, the jars and boxes used for the museum technique. The boxes are called perspex boxes, which are lab-made or commercial. The glass jars and boxes are also used. The specimens are attached sometimes to center plate. In the plate, the specimen is placed in proper anatomical position. In which part of the particular organ we want to show, accordingly this thing has been done. Stitch the specimen to center plate with nylon or linen thread. Center plate with specimen is next fixed in the box. And in a particular specimen, we can show it according to the view we want to show. The boxes filled with the mounting fluid, and it is filled 1 centimeter above the specimen height. Remove any air bubble. Seal the top of the box with perspex cement, and seal the glass jars also with asphaltum rubber compound. So in this way, the specimens are stored in the jars and boxes. The basic museum techniques. It involved number 1, reception, number 2, preparation, number 3, fixation, number 4, restoration, number 5, preservation, and number 6, presentation. The basic thing is the reception of the specimen, not only for the museum techniques but also for any kind of histological tissue preparation. We first receive the specimen. Any specimen received in the museum should be recorded in a reception book. This number, which is given in the reception book, will stay with the specimen even after it is catalogued in its respective place. The reception book should contain all necessary information about the specimen, like clinical, gross, and microscopic findings. After the reception process, the preparation of specimen will be started. An ideal specimen is received fresh in condition and of course in the unfixed state. However, it is mostly obtained from pathological laboratory. After being examined, thus will already be formalin fixed. If the planning to use a specimen for the museum, part of it can be kept without disturbing for the museum. For example, in kidney, it can be bisected and one half kept aside for the museum. So any specimen, if it is a brain, kidney or any kind of organ of our body, it can be bisected or sectioned according to the demonstration purpose, and they can be placed for the museum technique. In the preparation, the specimen can be obtained from autopsy or they directly obtained from the operation theatre. So, the specimens are first cut according to the need of the museum technique, and they are cut by the brain knife or butcher's knife.